Benjamin Lewis. Benjamin joined King's College London in 2019 to begin his postgraduate studies. He works on investigating the AcrAB-TolC multidrug efflux pump, probing the structural dynamics of the system through mass spectrometry methods and other biophysical techniques. So looking forward to your talk. Thank you.
Thank you very much. So yeah, as said, I'm Ben from the third year PhD from the Reading group. Of course, I don't have to tell everyone that antibiotic resistance is rising at an incredibly alarming rate, hence why we're all here. But there's also this problem of multi-drug resistance, and efflux pumps are a huge driver of multi-drug resistance. Not just the fact that these efflux pumps exist, but under stress conditions, they're overexpressed and they can efflux a wide variety of antibiotics at an incredibly quick rate. And this makes infections incredibly hard to treat because a whole host of antibiotics will be rendered ineffective. So specifically, we look at the AcrAB-TolC multi-drug efflux pump. So this is native to E. coli. It's made of 3 proteins, AcrB, inner membrane protein, AcrA, membrane fusion protein, and TolC, outer membrane protein. We study the AcrAB-TolC pump because it's quite well characterized in terms of the fact that there's a cryostructure of the assembled pump, the efflux mechanism is known and documented, and there are homologs of this pump across other escape bacteria. So the results and findings from this system transfer quite nicely. That being said, there is quite a lot that isn't known about this pump, such as the assembly mechanism and conformational dynamics of the pump. I do want to touch upon the rotational mechanism before I get onto the rest of the talk. So AcrB, which is the inner membrane protein, and represented by these shapes here, these colored shapes, cycles through, it's a trimer, and each monomer cycles through 3 different states. Loose, when the drug binds, tight, when the drug is moved into a tighter binding pocket, and then open as the drug is released into the channel of the assembled pump, like so. And there's a lot of conformational changes within AcrB as it cycles through these 3 different states. But if you then think about AcrA, represented by this pale green shape, this protein also has to be incredibly flexible, incredibly dynamic to actually, contain the conformational states of AcrB within the assembled pump. And that leads us to our aim of targeting AcrA. Now, the Segurtzia group in University of Oklahoma, who we collaborate with, identified NSC 60339 as an AcrA inhibitor. So they also did a proteolytic digestion assay which suggested that this causes a conformational change within AcrA, and this is an efflux pump inhibitor. However, there's no structural biology information available, so the mechanism of inhibition isn't known, and the where it is impacting AcrA, it isn't known either. So we want to use hydrogen deuterium exchange mass spec on this system to try and tease out some of this information. They also identified another biosynthetase control, which is an aminocoumarin antibiotic that binds to AcrA but doesn't seem to cause a conformational change. So a slight touch on hydrogen deuterium exchange mass spec for those that aren't too familiar with the technique. So basically, we're following a chemical reaction, which is a backbone amide hydrogen exchange for heavier deuterium. And we measure this change and this reaction via mass spec. So this reaction is dominated by 4 parameters: temperature and pH, which we control, then solvent accessibility and hydrogen bonding. So really the rate of uptake and exchange gives us a great idea on backbone dynamics. So what we do is we label our protein in solution in deuterated buffer for various time points, ranging from, say, 10 seconds to 4 or 5 hours. We quench this reaction, so we bring it to these parameters here, because the reaction is reversible and we don't lose any information. Our protein's then digested, the peptides ran over an LC, and then we can measure the rate of exchange via the mass change from deuterated to non-deuterated. And then we can perform statistical analysis to make sure our results are valid. So we can have a first look at AcrA dynamics, and this is the first kind of structural information on AcrA dynamics available. And I kind of want to highlight one thing here. So on the left, we have a heat map. So this is the deuterium uptake for all our time points, as you can see here. So as you go down, the time points are increasing. So this is top will be 10 seconds, at the bottom will be 4 hours. So I want to highlight the areas in red at 10 seconds. So if we have maximum uptake after just 10 seconds, this is indicative of unstructured regions, because the protein structure has not been able to settle on any secondary structure. It's completely solvent accessible and lacks the secondary structures, or lack local hydrogen bonding and stuff. And then we can look at the structure on the side here, which is the data for the 10-second time point. And we can see across actually all 4 domains, there is unstructured regions and rapid uptake at just 10 seconds. So we thought to investigate this further with native mass spec. And very briefly, this just involves transferring native folded proteins in solution to the gas phase, and then that can be monitored by mass spec. So we have our 2 native mass spec for AcrA at pH 7.4 and pH 6. They look the same, which is good. And it's both a monomeric, which we expected. However, there's 2 charge state distributions. There's a lower charge state distribution, as you can see here, which is indicative of folded protein. But then there's also this secondary distribution at a higher charge state. And this is a characteristic of a protein that contains intrinsic disorder. And that's what I want to highlight here. So this native mass spec is supporting our hydrogen deuterium exchange data in that AcrA, it contains areas of intrinsic disorder actually across all 4 domains. Then we can look at our NSC 60339 efflux pump inhibitor. So this is differential HDX. So for the latest time point, this is the delta HDX loaded onto the protein structure. So this is just AcrA plus drug minus AcrA on its own. So we're seeing the effects of the drug on this rate of exchange. And what we can see is a stabilization across multiple regions of the protein. So the rate of exchange decreased in the presence of drug. And what the results are suggesting is that the drug is actually binding between this lipo and alpha beta domain. And this results in a stabilization across these two domains, quite significant stabilization. And if you then think back to that rotational mechanism, what this could be doing is actually acting like a bridge or a staple between the 2 domains, restricting AcrA's conformational flexibility. Hence, efflux is less effective as AcrA struggles to adapt to AcrB's transition between its 3 states. There's also stabilization in the membrane proximal domain, which could either be a secondary binding site or a drug-induced conformational change downstream. We do know that the drug inhibits this protein when it lacks the membrane proximal domain, supporting our idea that this is where the drug is binding. And here is just a closer look at the binding sites. The residues I've shown were indicated in simulations to actually interact with the drug as well. And you can see statistical hits here for backbone dynamics anyway in our data. We can compare this to novobiocin, which paints a very different picture. Our AcrA dynamics is pretty much unaffected by novobiocin, as the whole protein is pretty much colored white, meaning there was no significant change, which is what we would expect, really, because it wasn't. Previous preliminary studies suggested it didn't cause conformational change. There is a slight increase in exchange in this left-hand helix. It might be due to more unwinding events, etc., etc. But overall, it's not having anywhere near the same effect as our inhibitor. So relating it back, what our key findings, basically we're able to capture the first captures of AcrA dynamics, and it reveals areas of intrinsic disorder supported by both our mass spec techniques. Our inhibitor looks like it could be bridging that lipo and alpha beta domain, restricting the conformational flexibility of AcrA as AcrB cycles through these 3 states. thus making the efflux rates less effective and less efficient. And our control number biasing has little effects on dynamics. So next steps, and what the next kind of year will look like, is actually we want to investigate AcrA as a pseudo homodimer. So we want to have two AcrA molecules linked by like a 5 amino acid linker, and we want to do this to basically, in the assembled pump, AcrA forms a hexamer, but it's more like a trimer of dimers, and we want to see if the dimerization would stabilize this intrinsic disorder, and then the impacts of drug or the two drugs on the dimer construct, and then also investigate the impact of the periplasmic environment on dynamics and pump assembly. So that's the presence of magnesium peptidoglycan mainly. Acknowledgements to the funders and everyone that's helped with this. So it's everyone in the Reading group and Booth group at King's and the Skier group at the University of Oklahoma. Thank you.